Hey everybody, welcome to the Heart lecture video. This is chapter 18 of the Marieb and Hoehn Human Anatomy and Physiology textbook. We are going to jump right in with the heart. So obviously the heart is anatomically one organ, but functionally it's actually two pumps that just happen to be located right next to one another. They would technically function if they weren't right next to one another because they're pumping, each one is pumping into its own separate circuit and receiving blood from a separate circuit. Now, those two circuits are continuous, but the idea is that these pumps are actually in series. So, you know, it's just obviously more efficient to have them located right next to each other. So they are, but functionally they are two separate pumps. So what are those two separate pumps? Well, we call them, it's very creative, we call them the right heart and the left heart. And each heart, in air quotes, is made up of an atrium and a ventricle. So the right heart is made up of the right atrium and the right ventricle, and the left heart is made up of the left atrium and the left ventricle. So pretty simple, makes sense. The atria are these smaller chambers on top. Let's see if I can draw on here. I can, yay. So these are atria. They are the receiving chambers. They receive blood from the pulmonary and systemic circuits, which we'll describe in a second. And the ventricles are the pumping chambers or the discharging chambers, and those pump blood into either the pulmonary or systemic circuit. So what are these circuits? So the pulmonary circuit is the part of the circulatory system that delivers blood to the lungs to be oxygenated. So it isn't supplying the lungs with oxygen. The right heart specifically is pumping blood into pulmonary circulation. It's hypooxygenated blood being pumped to the heart to pick up oxygen. Then that oxygenated blood comes back to the left heart where it's ultimately pumped out into systemic circulation, which is our system. It's delivering oxygen to the tissues of our body. So again, pulmonary circuit is up top in this diagram. It's a part of the circulatory system that takes hypooxygenated blood to the lungs and returns oxygenated blood to the heart. And the systemic circuit takes or delivers oxygenated blood to all of your body's tissues with the blood supply, and that hypooxygenated blood then returns to the heart. So there is a little bit of a trick question that I'll put on a quiz sometimes, so I'll just bring it up now. And it is, the left heart delivers blood to all tissues of the body that receive blood or that have a blood supply, or all tissues of the body that have a blood supply except for the lungs. And the answer is actually A, because the systemic circuit supplies all of our body's tissues that have a blood supply with oxygen, even the lungs, even cells in the lungs need oxygen. The difference is the pulmonary circuit is taking hypooxygenated blood to the lungs, to the alveolar sacs, to pick up oxygen, to become oxygenated again. The left heart is pumping blood to the system, which is all of our body's tissues that have a blood supply, to deliver oxygen, nutrients, and pick up waste. So a subtle difference there. And then last thing for this slide, I just want you to know what each heart, where does it receive blood from, which circuit, and where does it deliver blood to, which circuit. So the right heart receives hypooxygenated blood from the systemic circuit and delivers that hypooxygenated blood to the pulmonary circuit to be oxygenated. The left heart receives oxygenated blood from the pulmonary circuit and then delivers that oxygenated blood to the systemic circuit, to the body's tissues, to supply them with oxygen. Hopefully that makes sense. We're going to go through that a lot more, so hang in there. Let's look at size, location, and orientation. So your heart is roughly the size of your fist. Now I've seen hearts that are much bigger in our cadavers, but that person probably had some sort of disorder that caused that cardiomegaly, like high blood pressure, for example, or heart disease. There are various things that could cause cardiomegaly, and having a big heart is not necessarily a good thing. In fact, it's probably a bad thing. A healthy heart is roughly the size of your fist and weighs less than a pound. It's found within the mediastinum. That's the area between the lungs and above the diaphragm. And the heart actually rests on the diaphragm. Now, when I go into lab, not doing that right now, of course, but when I go into lab, oftentimes we will remove a few hearts from the cadaver so you can see them. We do a lot on the heart models in lab, but we usually try to have like one heart available or two. Oftentimes we'll pick one of the bigger ones. Anyway, so what we would have to do is dissect that out. And there's a lot of fat around the heart oftentimes, not always, but oftentimes. And then the heart actually sits within this sac called the pericardium. And we're going to go through the layers of that pericardium right now. So the outermost layer of the pericardium is called the fibrous pericardium. And you can see in this picture right here, this is the fibrous pericardium. We're at the top of the heart where the great vessels exit, and you can see how it's adhered to the pulmonary trunk. So the fibrous pericardium, it's tough. It's made of fibrous connective tissue. It protects the heart. It also prevents the heart from overfilling. If you've ever sprinted and then all of a sudden sat down for some reason, that'd be weird, but maybe you did, you might notice some really big thuds. Boom, boom, boom. You might actually feel your heart thudding. And that's because your heart was just really cranking, right? Because you were sprinting. So your heart's beating as hard and as quickly as it can to deliver oxygen. Meanwhile, all of your muscles are contracting, so all of your big veins that are filled with blood are being squeezed out, and all that blood is rushing back to your heart. So you have a huge increase in venous return and cardiac output simultaneously. And then all of a sudden you stop running. Well, your blood is still moving fast and at high volume, but your heart is starting to slow down because that oxygen demand has gone away because you've sat down, you're not running anymore. So the heart starts to overfill dramatically and then thud to push that blood out. Well, one of the things the fibrous pericardium does is it kind of makes a boundary. It prevents your heart from overfilling, overstretching, and damaging itself. Below the fibrous pericardium, you have two serous layers and then the pericardial cavity in between. So this is a little out of order, I'm sorry. So you have the parietal layer of the serous pericardium, which is right there. Remember, parietal is like wallpaper, visceral is like gift wrap. So you have the visceral layer covering the heart, and then the parietal layer lining the cavity. So those are the two serous layers of the pericardium, the parietal layer of the serous pericardium, and the visceral layer. In between them, you have the pericardial cavity, which is filled with a serous fluid. It's like a blood filtrate. And its job, the job of this cavity, the cavity here, the pericardial cavity and the fluid within it, is to provide an environment that the heart can beat without friction or minimal friction. It's sort of floating in this fluid-filled sac and moving around as it contracts without rubbing anything. And that prevents tissue damage to the heart. I have another website where I make boards questions for people studying for the National Physical Therapy exam. And one of the questions I put on there, it's actually a super easy question when you have multiple choice, but it's kind of a hard question if you don't. And the question goes something like this. So how do you differentially diagnose between a pericardial friction rub and a pleural friction rub? Most simply. So I need to describe what those two things are. If you imagine that this fluid, maybe due to some sort of infection or other disorder, becomes extra viscous or there's less fluid than normal, it could start to rub and create friction, and that would be very painful to the patient, so they'd have chest pain. The same thing can happen in the cavity between the pleural membranes of your lungs, the visceral pleura and the parietal pleura. There's a very thin fluid compartment between those two layers. So when you breathe, you can experience chest pain. Now, granted, a pleural friction rub is going to be more widespread and diffuse, and a pericardial friction rub is going to be, right near your heart. But they can mimic one another. They can feel similar. So how do you differentially diagnose? So if it's A, B, C, and D, it's a no-brainer, but it's harder if it's fill in the blank. So what would you do? Some of you might be shouting at the screen right now. I don't know. But let me answer the question with a question. Can you ask a patient to temporarily stop their heartbeat? You can, but they'd probably give you a funny look. That's not, they're like, hey, I came to the doctor. No, you can't. But you can ask a patient to temporarily stop their respiratory cycle. You can ask them to hold their breath. So if the patient holds their breath and the pain continues, it's most likely a pericardial friction rub because the heart's beating but the lungs aren't moving. So you've temporarily stopped any chance of there being a pleural friction rub because the lungs aren't moving but the heart is. If you ask the patient to hold their breath and the pain stops, then it's probably a pleural friction rub. That's a huge tangent, and I won't ask you any questions about that, sorry. But I just want you to know the order of these layers. If a needle is inserted in through the pericardial sac, through the pericardium, in what order would the needle pierce the layers? So fibrous pericardium, parietal layer of the serous pericardium, visceral layer of the serous pericardium. Let's look at the layers of the heart wall. I forgot to mention this layer here, the visceral layer of the serous pericardium. It's also considered to be a part of the heart wall. When we're talking about the heart wall, we tend to call it epicardium. When we're talking about the pericardium, we tend to call it the visceral layer of the serous pericardium, but they're the same thing. They're synonyms. So anyway, here we go. Layers of the heart wall. Epicardium, that's the outer layer of the heart wall, also called the visceral layer of the serous pericardium. Below that, we have the myocardium, which is just the muscle within the heart wall, the contractile tissue. In lab, we talked about how there's different names for different parts, trabecular carne, pectinate, papillary muscles, but all this contractile tissue is a part of the myocardium, in addition to a non-contractile skeleton called the cardiac skeleton. This skeleton is not calcified, but it's not bone, it's just connective tissue, but it gives the heart muscle something to tug on, and it gives the heart some structure. So it's very important. And then lining the chambers of the heart and all vessels of the entire circulatory, well, let me back up. Lining the vessels of the heart, the chambers of the heart, is endocardium. This is an endothelial sheet. It's continuous with the endothelial sheet that lines your entire cardiovascular system. So all of your vessels have an endothelial sheet. This is the inner layer of an artery. Some capillaries are composed only of endothelial cells, and these are cells that release various chemicals that manage inflammation and blood pressure and stuff, so it's important. Disorders like atherosclerosis can damage it, and that's not good. But anyway, endocardium is the innermost layer of the heart wall and is continuous with the endothelial sheet of your entire vascular system. Okay, I think you guys could do both of these, right? Oops, I didn't mean to cross it out. So you know the order of the layers and what's the purpose: to reduce friction and to minimize damage to the heart due to friction. Okay, let's look at vessels and chambers. I only wrote chambers on here, but it's titled chambers and vessels, so let's name all of them. So this would be the right atrium. This would be the right ventricle. The right ventricle pumps blood into the pulmonary trunk. That blood comes back into the heart via the pulmonary veins. They're called veins even though they're carrying hypooxygenated blood, which is unusual for a vein. We think of veins as carrying hypooxygenated blood back from the body, from the system. These are veins carrying blood back from the lungs, rich in oxygen. So the definition of a vein is a vessel carrying blood towards the heart, and an artery carries blood away from the heart. So anyway, those are the 4 pulmonary veins. They deliver oxygenated blood to the left atrium. Left atrium pumps blood into the left ventricle. Left ventricle pumps blood into the aorta. It's supposed to be an arrow. This is the aorta. Okay, and on another slide we'll actually go through the details of the blood flow pattern. I just wanted you to see those 4 chambers and the great vessels. Oh, I forgot to mention the vena cava. These are 2 vessels bringing venous blood back from systemic circulation into the heart. We'll talk about all that in a second. I just want you to know their names and their locations. So 2 atria, 2 ventricles, and then the various great vessels. Let's look at the valves. So this is the front of the heart. So this would make this the left AV valve right here and the right AV valve. AV stands for atrioventricular. So these are valves between the atria and the ventricles, left and right. Kind of an older name for the left AV valve is mitral valve, and the right AV is tricuspid valve. It just depends who you're hanging out with. Some people like jargony words because it kind of makes them sound cooler. Honestly, I think that's all it is. Maybe not. But a lot of the different anatomical societies that come up with names for things are trying to move towards simple verbiage that's a little bit more explanatory, so less mistakes are made, and there's less confusion. Anyway, so that's left atrioventricular valve is the newer and more common name for the mitral valve. If you're doing like a residency with a cardiologist or something, I'm sure they'll call it the mitral valve. Old habits, but anyway, left AV, right AV, and then you have the aortic semilunar valve and the pulmonary semilunar valve. So those are the 4 heart valves. And we're going to talk all about what they do, which way they open, what pressure gradients have to exist for them to be open or closed. But we're just getting started. This will probably be a longer video, so hang tight. Pathway of blood through the heart, page 669, important. Unfortunately, I don't have the image here. So what I'm going to do is I'm going to go back here, and we're going to walk through the pathway of blood flow through the heart. Now, I'm going to write numbers, but I will not ask you like, hey, what is step 1 of the blood flow pathway of the heart, or step 2? I'm just doing this for myself to keep track. So step 1 would be, let me back up a second. Let's pretend that we are on the magic school bus with Miss Frizzle. We all got shrunken down to the size of a red blood cell. We were put in a syringe and we were injected into a vein. If we were injected into a vein above the heart, we would enter the heart through this vessel right here, which is called the superior vena cava. If we were injected into a vein below the heart, we would enter the heart through the inferior vena cava. So the vena cava bring blood back to the heart from systemic circulation. So venous blood from systemic circulation is delivered to the heart through a vena cava. If it's in your upper body, it'd be superior. If it's in your lower body, it'd be inferior. So that's step 1 of the vena cava. Step 2 would be the right atrium. Step 3 would be the right AV valve. Step 4 would be the right ventricle. And these arrows are kind of showing that pathway, right? 1, 2, 3, 4. Step 5 is the pulmonary semilunar valve. Step 6 is the pulmonary trunk. Step 7 is one of the pulmonary arteries. The pulmonary arteries deliver hypooxygenated blood to one of the lungs. So you have a left pulmonary artery and a right. So that blood goes to the lungs. That comes back through one of the 4 pulmonary veins, which you guys know from lab is right superior, right inferior, left superior, left inferior pulmonary vein. All 4 of those empty into the left atrium. Blood then passes through the left AV valve into the left ventricle, through the aortic semilunar valve into the aorta, which is the start of systemic circulation. Once this valve opens, the aortic semilunar valve, that's when you would feel your pulse at your wrist or anywhere, because all of a sudden your arterial pressure increases about 40 millimeters mercury when that valve opens. All right, so that was a quick run through. More than anything, I want to know, like, you might see a test question like, okay, so if you just passed through the right AV valve, which chamber are you in? Well, if I just passed through this valve right here, the right AV valve, that must mean I'm in the right ventricle. Those types of questions. Not, what is step 6 of the pathway of blood flow through the heart? All right. I think I'm going to take a quick break. Be right back. Okay, so now we're going to look at, we're going to go from looking at kind of a gross anatomy overview of the heart to looking at the microscopic anatomy and the physiology of the muscle fibers and the action potentials and mechanisms of contraction and all that kind of thing. So let's jump into microscopic anatomy. I think I meant to have this be like separated here in boxes, I'm sorry. But anyway, we're going to compare and contrast on the next few slides, I think, some of the differences and similarities between cardiac muscle tissue and skeletal muscle tissue. They're two different types of tissue. Although they're both muscle and they both contract, they're very different in the way that they work, the way that they function. So cardiac muscle is short and fat, and all of the fibers are physically and chemically interconnected. That's why they're actually not called fibers. We said cardiac muscle fibers back here. I think I pulled that from your book. But typically we do not refer to cardiac muscle cells as fibers, but you'll see it. It's hard to just make an exception for that one type of muscle. But anyway, they're short, fat, and interconnected, whereas skeletal muscles are long and cylindrical. Kind of a similarity almost here. Skeletal muscle is connected to the skeleton via connective tissue, and the cardiac muscle is connected to a fibrous cardiac skeleton via connective tissue. So both utilize kind of a semi-rigid or rigid structure, and the fibers or the cells are connected to that structure using connective tissue. Here is the big difference. This is key right here. Muscle fiber, skeletal muscle, this is from last quarter, is functionally and electrically interdependent. What that means is, say my biceps muscle has 100,000 skeletal muscle fibers. I can turn on specific ones or groups. I can turn on 10% of the muscle. I can contract 40% of the muscle to adjust how much force it's producing to lift something that's light or to lift something that's heavy. The heart does not work that way. The heart cells, the muscle cells, are interconnected by something called an intercalated disc, which is a special type of gap junction, which, if you remember from last quarter, is just basically a tube or a tunnel between adjacent cells so that the fluid within each cell and the ions within each cell are able to move between cells. So it's basically just a tunnel between cells. But what this means is that if one muscle cell contracts, the electrical signal, the action potential within that muscle cell is naturally going to spread to the adjacent cells. So the heart is kind of on or off. It's not 10% contraction, 50% contraction. It doesn't produce graded muscle contractions the same way that skeletal muscle does. Lastly, this is kind of an average based on different fiber types, but skeletal muscle is about 2% mitochondria by volume, whereas heart muscle is 25 to 35% mitochondria by volume. This is because heart muscle tissue utilizes aerobic respiration to a much greater degree than the other muscle cell types, because you do not want your heart to fatigue. Heart fatigue is not good. You want a constant, continuous supply of energy using aerobic respiration. Remember, anaerobic respiration is limited in the amount of ATP it can produce and how long you can rely on it for ATP production. So you want aerobic respiration that's very efficient, producing a lot of ATP so that your heart continues to function normally. This is just a quick diagram of skeletal muscle tissue up here. It's a single long fiber versus cardiac muscle. It's shorter and fatter, and they're interconnected. And there's intercalated discs right here. They're showing you that connect one cell to another. So these are two different cells, and they're connected by gap junctions, and they're also physically connected. So that brings us to this. So based on everything we just learned, how do we apply that to the actual events of contraction? So there are three fundamental differences between cardiac and skeletal, and we've covered this a bit. So this one we've covered for sure, so let's start there. The heart utilizes organ contraction. It's all or none. The heart's either contracting or not. Skeletal muscle tissue uses motor unit contraction, which we talked a lot about last quarter. I won't ask you for specific questions like what is motor unit contraction, because that was last quarter, but I will ask you to differentiate between cardiac and skeletal. So you need to know it in that extent, but don't worry if you weren't here last quarter, that's not going to affect you. But anyway, in skeletal muscle, impulses do not spread from cell to cell. Each cell is isolated electrically from one another, so your nervous system can actually decide which cells are contracting and which aren't. It's not the case for the heart. Which brings me to, how is the heart stimulated? Well, it's self-excitable. We're going to learn in a second that there's a special subset of cells within the heart that are autorythmic, meaning they depolarize at a predictable rate. That rate can be influenced, of course, but they're auto-excitable, they're self-excitable, meaning that you don't actually need the nervous system to tell the heart to contract. The autonomic nervous system can speed it up or slow it down, but even if you were to, like in a vampire movie, pull a heart out of somebody's chest, it would keep beating till it ran out of ATP or calcium or whatever other thing it needs to beat. That's why a heart transplant works. You don't necessarily have to reconnect neurons. The heart will beat on its own. After a heart transplant, though, your heart rate will be higher, and we'll learn why that is later. But anyway, the point is, cardiac muscle tissue is self-excitable. Skeletal muscle tissue is innervated by the somatic motor nervous system. So each skeletal fiber is connected to a nerve ending which tells it whether or not to contract. And then lastly, and also very important, is the length of the absolute refractory period. Skeletal muscle absolute refractory period is very short. That allows you to flex a muscle because it's being stimulated just rapidly. Every 1 millisecond, there's a new there could be a new action potential potentially, and that would cause contraction of the muscle cell. That's not the case in the heart. There's more than a 200 millisecond period where the heart cannot contract again. This prevents the heart from flexing. You don't want the heart to contract and flex like a biceps muscle because then blood can't get in. You want it to relax to allow it to fill with blood and then contract. So this refractory period, it almost kind of sets a max heart rate because there has to be at least a 200 millisecond gap between beats due to this absolute refractory period. This is very important. So there's a longer absolute refractory period in cardiac muscle than in skeletal muscle. Okay, man, these slides are crazy. I switch between Word and Apple a lot, PowerPoint and Keynote a lot, and it creates all kinds of problems, so I apologize. I need to stop doing that. But anyway, so now we're going to be talking about action potentials of cardiac muscle. So this picture right here is what the action potential of most, about 99% of cardiac muscle cells looks like. A very steep depolarization phase. Look at that. That's unreal, right? It's just almost an instantaneous depolarization phase, very sharp, with a long repolarization phase. That long repolarization phase creates a long absolute refractory period. So the absolute refractory period is from here to here. The green line is tension production. So that's the contraction itself. So you see you can't really make another heartbeat until the heart is almost fully relaxed, or at least halfway relaxed. So that creates, the heart has to relax a little bit between beats. If it were to flex, that's not good at all. Because there'd be no blood flow. It'd be similar to a heart attack. 1% though, 1% of cardiac fibers are autorhythmic. So they display something called a pacemaker potential, which we will explain in a few slides. But the point is that there's 1% of these muscle cells that generate action potentials on their own. And because the heart is interconnected electrically and physically, that action potential spreads. So there's a pacemaker in your heart that sets the pace. Don't think of like an implantable pacemaker. That's something that's put in artificially. I have an interesting story about a pacemaker actually. So I was in the cadaver lab one time by myself, and it was dark, which is never a good idea. No matter how logical and rational you are, you get in a room filled with 60 cadavers and you're alone and it's dark and you start spinning every time you hear a noise. But anyway, oh, that reminds me of another funny story too, but okay. So I'm sitting there and I hear a beep, 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 beep. And I'm like looking around like, what is that? And then I go back to work. I think it was dissecting a hand or something. Beep, 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 beep. And I'm like, this is coming from like this table, and beep, 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 beep. And I think it went on for like a week. Every day in class we would hear it. And finally, when we were dissecting out the heart, we realized that this particular cadaver had a pacemaker and the batteries were low in the pacemaker, so it was beeping. Anyway, it was just kind of crazy, which reminds me of another story, which is very unprofessional, but there was once a student, this was years ago, who put like the tarp over himself on a gurney, on a new gurney on a cadaver table. So someone walked up to push it and he sat up and gave everybody a heart attack. So that was not very funny. Anyway, sorry. So 1% of cardiac fibers are autorythmic. Let's talk about what that means on the next slide. First, we need to address that 1%. What does it do? That 1% of cells that are autorythmic. Well, that's a part of the heart's intrinsic conduction system, meaning the heart has a conduction system within the heart. It doesn't need the nervous system to initiate contraction like our skeletal muscle does. And that this independent activity of the heart is due to that 1% of muscle tissue, of the muscle cells within the heart that are non-contractile conductors and that are autorythmic. And then second, or in my list first, the presence of gap junctions. So we have cells in your heart that generate action potentials, and then because of gap junctions, those action potentials spread. So let's look at the action potential initiation by pacemaker cells. So these cardiac pacemaker cells, again, broken record, are autorythmic. Unlike neurons, though, they have an unstable resting potential. We call that unstable resting potential a pacemaker potential. So if you look at this list, these are two action potentials. So these are the peaks of two different action potentials. So they kind of look like a neuron action potential. You have depolarization, you have repolarization, you have hyperpolarization, right? There's one thing that's different about these. There is no resting membrane potential. Or I guess we can say there is a resting potential, but it's unstable, it's sloped. So what that means is that these cells are going to automatically hit threshold no matter what because of the sloped resting potential. And that sloped resting membrane potential is called a pacemaker potential. And it's just a slow depolarization event. And then you reach threshold, you get depolarization, repolarization, hyperpolarization, and then pacemaker potential. Depolarization, repolarization, hyperpolarization, pacemaker potential. So because of this, you get a routine depolarization that happens on its own autorhythmically. And again, these are called pacemaker potentials, that sloped resting membrane potential. So what does that do? Well, all of this 1% of cells, which are highlighted in yellow in this diagram, display a pacemaker potential. And the different parts of this intrinsic conduction network, again highlighted in yellow, have pacemaker potentials with a different slope. So each part of this system would generate action potentials at a different rate. For example, right here, the SA node, it has an action potential on its own about 75 times per minute. The AV node is about 50. The rest is somewhere between 50 and 30. Now what's interesting though is that because once an action potential is generated, it spreads. Whichever part of this network is generating action potentials most quickly sets the pace for the entire heart. So in a healthy heart, the SA node is the heart's pacemaker because it produces action potentials most rapidly, about 75 times per minute. So right now in your heart, the SA node is depolarizing 75 times per minute, and that's setting the pace of your heart. Now, most of us, our parasympathetic nervous system will slow that rate down a little bit to about 60. But a normal heart rate is about 60 to 90, 60 to 100, so it's going to be somewhere in there. We just say 75, which is kind of an average. But anyway, that's the SA node. So the AV node, it doesn't matter that it beats 50 times per minute, depolarizes 50 times per minute. It doesn't matter that the Purkinje fibers depolarize 30 times per minute because the pace of the whole heart is set by the most rapidly depolarizing region, which in a healthy heart is the SA node. So if there's a question on the test that says, in a healthy heart, what structure paces the heart? The answer would be SA node. Now, if something were to happen to the SA node, honestly, but before we do that, let's look at the conduction pathway, the sequence of excitation. Then we'll come back to that. So we know the action potential is going to initiate from the SA node, so that's step one. The sinoatrial node generates an impulse. That impulse spreads, that's what these black arrows are, throughout the atria. Now you might say, well, why doesn't it spread to the ventricles? Well, this septum here, which we learned in lab is called the atrioventricular septum, is insulatory. So this action potential can't spread down through the valves or through the septum. It's cut off. It has to go through the AV node. See that in order to get into the ventricles, that action potential needs to go through the AV node. But the AV node generates a pause, a 0.1 second pause. So that allows this impulse to spread throughout the atria, which is why the atria contract before the ventricles. And that's very important that they do that. But anyway, one of these, not one of these, but that spread of action potential will eventually reach the AV node. The AV node creates a 1 second pause, and then it allows the action potential to spread down through the AV bundle. All right, the AV bundle connects the atria to the ventricles. So that's step 3. And then it spreads through the bundle branches. These are new names. You might see names like bundle of His, and there's a bunch of different names, but these are the current names. Well, 2011, but these are the current names. And then after the bundle branches, it goes into these littler structures called Purkinje fibers. So you can see the pathway of excitation, the action potential spread along this pathway. And this is how the contraction will look as well, because remember the electrical event, the action potential triggers a contraction. So we see the atria contract first, then we see the septum contract and the apex move up, pushing blood into these valves here, and then you see the walls kind of go in. So it's forcing the blood out and squeezing the blood out through the great vessels. So that is the path of contraction. I wonder if this is a good spot. Not really, but I just want to show really quick that this idea of how the actual pumping of the heart works. So the atria are pumping the blood into the ventricles. When the ventricles contract, it shuts these valves right here because these are one-way valves. And the ventricle contraction forces blood into the aorta and the pulmonary trunk, which they don't really have drawn here. We're going to talk about this a lot in the cardiac cycle, so don't fret. We have 2 centers within our medulla oblongata that can slow down SA node depolarization and AV node depolarization, or speed it up. So the sympathetic system, utilizing the cardioacceleratory center, can speed up heart rate, and the cardioinhibitory center can slow down heart rate. Most of us have something called parasympathetic tone, which means that our parasympathetic division is sort of winning that tug of war. So our SA nodes are slowed down slightly from their baseline depolarization rate, which is about 75 times per minute. Okay, let's go on to the ECG. Have you guys ever heard of an EKG? Do you know what the difference is between an EKG and an ECG? They're not different at all. They're the exact same thing. It was invented in Russia, I believe. I don't know if it was invented in Russia or popularized in Russia, but either way, they spell cardio with a K. So it was an EKG, electrocardiogram with a K. We say ECG because we spell cardio with a C. But anyway, ECG. An ECG is basically the summation of electrical events of the heart throughout one cardiac cycle. It's not an action potential. So it's whatever electrical information is reaching the electrodes that are spaced, all over your chest. And then what we see is that some electrical event. So we can't necessarily say like, oh, this means this means that, but there are certain generalizations we can make about certain electrical, certain graphical representations of electrical events and how that correlates to electrical events within the heart. So we'll run through this really quick, and then I'm going to take a quick break because we're getting ready to go into the cardiac cycle, which is a bit confusing, to say the least. So anyway, here we go. So you can see some letters up here. So this letters correspond to certain parts of the ECG. So this P corresponds to the P wave, which is this little bump right here. QRS complex, and then the T wave. And then there are other things which aren't necessarily on here that we'll look at, like the PR interval, which is the distance between the P wave and the QRS complex, or the ST segment, which is this line here between the S and the T. We can look if this is elevated or depressed, or if the PQ interval is longer or gets progressively longer with each beat. That those mean things. We can say, oh, it's a first-degree heart block or a second-degree heart block. We're not going to go into those, unfortunately. I'm sure you will, depending on what you do next after this class. But I just want you to know the segment so that when you get to that class, you kind of have a leg up. So anyway, the P wave corresponds to atrial depolarization. So it's if you see a P wave, that means that the SA node successfully depolarized and that spread throughout the atria. Then there's a little gap between the P wave and the QRS complex, and that is this period where nothing really is happening because there's a delay by the AV node. Now, if that segment was really long, we might suspect some type of heart block, meaning that the AV node is not functioning properly. We may even see dropped QRS complexes. And there's multiple types of heart blocks, first degree, second degree, third degree, and then within the second degree there's a Mobitz type 1 and a Mobitz type 2. So there's a lot of different types. We won't go into that, but just know that's an important segment. And that segment corresponds to the function of the AV node specifically. Okay, the QRS complex represents ventricular depolarization. Ventricular depolarization. Now, one thing this might be helpful for, of course, we have digital machines that do all this now, but if, let's say you took a 6-second ECG strip. Okay, so I picture you have one of those old machines with the pen, not the pen, but the little arm with the needle that's writing and the paper spins by it, right? And it's like an earthquake machine, a seismograph, or even just a digital 6-second frame of a digital ECG, and you count 6 QRS complexes. What is that person's heart rate? Well, it's a 6-second strip. There's 60 seconds in a minute. So if you multiply a 6-second strip by 10, that's 60 seconds, right? And so if each QRS complex corresponds to ventricular contraction, that means it corresponds to 1 heartbeat. So if you multiply the 6 QRS complexes by 10, you get 60 beats per minute. So in a 6-second strip, you would just multiply however many QRS complexes you have by 10 to estimate the heart rate. And I might put something like that on the quiz or test. Okay, that brings us to the ST segment. Not much is going on here. This is ventricular depolarization is complete, and it's a pause before the next phase, which is ventricular repolarization. So the T wave is ventricular repolarization, and then after that, ventricular repolarization is done, and it starts all over again. So the only thing we're missing on here is atrial repolarization. Atrial repolarization, you can see in red here, happens during the QRS complex. So it's kind of obscured by the massive ventricular depolarization event. It's kind of in there somewhere. We can't isolate it out. ST segment is crucial in diagnosing myocardial ischemia and infarction if it's elevated or depressed or sloping. The T wave is also involved in that. I'm not going to really get into those specifics for our class on the test or the quiz, okay? But I am going to ask you, what does the P wave represent? What does the QRS complex represent? What does the PR interval represent? You know, that kind of thing. I'm trying to think of some other ideas. Okay, if the okay, this is a good one. Do you remember how many times per minute the AV node depolarized? 50. But because the SA node is faster, the whole thing happens at about 75 times per minute. But let's say the SA node is damaged. The AV node would take over, and now the heart would beat 50 times per minute. Okay, so let me ask you a very difficult question. The P wave is absent. Okay, you're looking at a patient. The patient has no P waves. How many QRS complexes do you expect to see on a 6-second strip? You have all the information you need. It probably doesn't feel like it. Probably like, what? But you do have all the information you need. So let's break it down. If the P wave is absent, what's wrong with the patient? The SA node is not functional. So this is not a normal sinoatrial rhythm. It's not a healthy heart. The SA node is no longer functional. It's no longer the pacemaker. Well, if the SA node is no longer the pacemaker, what is? The next fastest part of the conduction system, which is the AV node. The AV node depolarizes about 50 times per minute. So how many QRS complexes would you see on a 10-second strip? In that case, you'd see 5. In a normal rhythm, a normal sinus rhythm, where the SA node is functional, you would see 6 or more, 7, 8, somewhere in that range, depending on the patient's heart rate. So I guess technically we should say 75 because that's the number we're using. So you'd see 7.5 QRS complexes in a normal sinus rhythm where the heart is paced by the SA node. But if the SA node is non-functional, there's no P wave, we would see a QRS complex, we'd see a heart rate of about 50 or lower because the AV node is now pacing the heart. Heart sounds. Lub-dub, lub-dub. It does actually kind of sound like lub-dub. Not at first, but when you start listening with the stethoscope, especially like a cool one that can record and you can play it back. Littman is kind of, I believe it's Littman. I should know this, but there's one stethoscope brand that everyone thinks is very good, and it is. And you can record. There's stethoscopes that you can record and do all sorts of things. But anyway, the two heart sounds are referred to as a lub and a dub. More scientifically, S1 and S2. S1 is the lub, and it's caused by AV valve closure. So you might think the noise your heart makes is due to contraction or something. It's not. It's due to valve closure. When the valves close, they make a noise. So when the atrioventricular valves close, that's S1. When the semilunar valves close, that's S2. So let's go back to this photo right here. So what would cause these valves to close, do you think? Well, let's figure it out. So if the ventricle pressure increases more than the atrial pressure, look at this valve. Which way does it open? It opens this way. So blood can flow like this. That's true of both AV valves. The right AV valve is just more visible. Oh, I guess not. So there we go. So blood rushes in as long as atrial pressure is higher than ventricular pressure. But if the ventricles contract, that situation reverses. Then the ventricular pressure is higher, so that closes this valve. It doesn't pump blood into the atrium because this is a one-way valve. So ventricular pressure increase, ventricular contraction, shuts the semilunar valves. Shuts the atrioventricular valves. So that's what causes your first heart sound. Ventricular contraction slams the AV valve shut. And then as ventriculars contract more and more and more, the pressure builds, it will open the semilunar valves, which is what you see in this picture. The situation in this picture would never exist. You would never have pressure leaving the ventricles and entering the ventricles like this. So this is kind of a bad diagram. But anyway, so the ventriculars are contracting. So it slams the semilunar, I keep saying that. It slams the atrioventricular valve shut and opens the semilunar valves. But the ventricles can't keep contracting forever. Eventually they have to relax. And when they relax, the aortic pressure pushes back and shuts the valve, just like the pulmonary trunk. As pressure decreases in the ventricles, the semilunar valves shut, and that's the second heart sound. And as ventricular pressure keeps lowering and lowering and lowering, eventually the AV valves open back up and the heart fills with blood. The ventricles fill with blood. So whether these valves are open or closed depends on the status of the ventricles. If they're contracting, that slams the semilunar, I keep saying that, I'm so sorry. As the ventricles contract, it slams the atrioventricular valve shut, that's S1. They then open the semilunar valves. As the ventricles start to relax, the semilunar valves shut, and that's S2. So we're going to do a better job explaining that on the cardiac cycle, which is next. So let's do that right now. Okay, so cardiac cycle. I show this little graph here or diagram, whatever you want to call it. And sometimes I wonder if it makes things more confusing or less, but I think it does a pretty good job of something that is borderline impossible, which is tying together all the different events of the cardiac cycle into one image. So ECG, heart sounds, pressure, volume, cardiac cycle, valve state. It ties everything into one diagram, and it is therefore very confusing, but it is also really the best way I've seen of describing all of these things simultaneously. So very simply, the cardiac cycle is composed of three phases: ventricular filling, ventricular systole, and early diastole. Probably a better way of calling this is mid to late diastole. You guys can't see that. Let me move the picture. For ventricular filling, because these are synonyms. Filling and diastole are really the same thing. So let me first explain systole and diastole, and then we'll go into this crazy image. So if you've ever taken a blood pressure or gotten a blood pressure taken, you get two numbers. You get a systolic blood pressure and a diastolic. So let's say 120 over 80, which would be a great blood pressure. That means that the blood pressure within your artery during ventricular contraction is 120, because systolic means ventricular contraction, or it just means contraction. And diastolic means relaxation. So 120 is the blood pressure when your ventricles are contracting, and 80 is your blood pressure when your ventricles are relaxing. Okay, so obviously there's going to be a pressure difference whether your heart is ejecting blood or not. Okay, so you can think of diastole as relaxation and systole as contraction. So really there's only two phases then, if you wanted to try to simplify it even further: ventricular systole and ventricular diastole. For whatever reason though, we split them up into early diastole and mid to late diastole, and there's a reason behind that, I suppose. But anyway, let's forget about that for a second, and let's make this as big as we can and go through this diagram. There is so much here, man. We could spend an hour on this, but we won't, I promise. Let's just right off the bat, let's just look at the phases again. So phase one, mid to late diastole. You can see that the ventricles are filling with blood, and the atria as well are filling with blood, but blood is moving from the atria to the ventricles. In the second part of mid to late diastole, we get some ventricular contraction, and the ventricles are topped off, and blood is forced from the atria to the ventricles. So that's step one, ventricular filling. Step two, 2A and 2B, is ventricular contraction. The first part of ventricular contraction, first of all, notice something. What's the difference between here and here? The AV valves are shut, and that's because the ventricles started to contract, forcing blood up, but because this is a one-way valve, the AV valves are shut. But so are the semilunar valves. The semilunar valves are also shut. And that's because aortic and pulmonary pressure is still higher than ventricular pressure. So ventricular pressure is in this weird spot. It's greater than atrial, but less than this aortic and pulmonary pressure. So all the valves are shut. So we call that an isovolumetric contraction phase, because the vessels are the ventricles are contracting, but all the valves are open. Then all of a sudden, ventricular pressure exceeds aortic pressure and pulmonary pressure, and the semilunar valves open, blood is ejected. That's called ventricular ejection. So isovolumetric contraction and ventricular ejection are both a part of ventricular systole. And then we get to early diastole, which means early relaxation. So the ventricles start to relax, but the pressure is still less than the aortic and pulmonary, but greater than the atria. So this is isovolumetric relaxation. All the valves are shut, but eventually ventricular pressure falls below atrial pressure and the ventricles begin to fill again. And then we start over with ventricular filling. Okay, let's look at this pressure portion and do that again. So here we are in ventricular filling. If you guys see that this phase here, like this is the first phase, and then second phase, and then third. Okay, so we're in the ventricular filling phase. So during the let me back up. So purple is atrial pressure, blue is ventricular pressure, and green way up here is aortic pressure. So we're just looking at the left ventricle in this case. So here we go. The left atrial pressure is greater than the left ventricular pressure. So imagine this is a test question. The left atrial pressure is greater than the left ventricular pressure. Is the left AV valve open or closed? Well, remember the left AV valve is like this. The atria is on top. All right, so here's the atria. The ventricle is on the bottom. So if this pressure is greater than this pressure, the valve would be open because it's a one-way valve that allows blood this way. If ventricular pressure went up, it would shut this valve. Okay? But right now, atrial pressure is greater than ventricular pressure, so the valves are open. But then something happens right here, ventricular contraction. The ventricles contract, ventricular pressure exceeds atrial pressure, boom, the valve shuts right there. Right when the blue line crosses the purple line, we get valve closure. And if we follow that up, there we go, there's the first heart sound. So the first heart sound, remember, corresponds to AV valve closure. So then we get this period where the blue is above the purple, so ventricular pressure is greater than atrial, but it's below the green line. So this region right here is going to be isovolumetric contraction. The ventricle is squeezing, but all the valves are shut, so the pressure builds. Then all of a sudden it crosses the green line, boom, we get ventricular ejection. The valves open, the semilunar valves open, blood is ejected. As long as the blue line is above the green line, the aortic semilunar valve remains open. But the ventricle can only contract so much, it starts to relax. And then the blue line falls below the green line right there. Boom, we get our second heart sound. We get semilunar valve closure. Okay. Then in this area we have isovolumetric relaxation. The ventricles are relaxing, but all the valves are shut. As soon as the blue line, the pressure of the left ventricle falls below the left atrium, the atrioventricular valves open and ventricular filling begins again. Okay, so that's kind of the pressure gradients that exist during one cardiac cycle. So be prepared for questions like, aortic pressure is greater than left ventricular pressure. Is the aortic semilunar valve open or closed? Well, let's look at it. So here's the aorta. It's a one-way valve that opens this way. Here's the ventricle. That's a bad ventricle because really there'd be another valve here and stuff, right? But anyway, so if ventricular pressure is greater, blood leaves. If atrial pressure is greater, the blood tries to move backwards, but it can't because it's a one-way valve that closes. Okay, so if aortic pressure is greater than ventricular, the aortic semilunar valve is closed. This is the same arrangement for the pulmonary trunk in the right ventricle. Oops. Let's look at that pressure thing again. And let's look at the electrical events and the heart sounds. So we're just going along here, atrial systole. What would we expect to see happen with atrial systole? The P wave. Because the P wave is SA node depolarization, which corresponds with atrial contraction and atrial depolarization. So those go together. If the P wave was absent, there would be no atrial systole. It's another good question for a quiz or something. Then we have the QRS complex. What does the QRS complex correspond to? Well, ventricular depolarization, which means ventricular contraction. So we see the pressure jump and cross the atrial pressure when the QRS complex happens. What else happens there? The first heart sound. As soon as ventricular pressure exceeds atrial pressure, we come down here and the valve shut. The aortic valve shut. Sorry, the atrioventricular valve shut. So the QRS complex corresponds with ventricular depolarization, which corresponds with AV valve closure, which corresponds with the first heart sound. So if I said which electrical event corresponds with the first heart sound, you would say QRS complex. So here we are, ventricular ejection. Okay, here, this is where the aortic semilunar valve opens, so we get ventricular ejection. Then the ventricles start to relax, ventricular depolarization. What electrical event corresponds with ventricular repolarization? That would be the T wave. So as soon as the semilunar valve shuts, we get the second heart sound. So I could ask you what electrical event corresponds with the second heart sound, and you would say the T wave. So this all goes together. Sorry, my wife was texting me. Okay, let's look at volume. So we have these terms, end diastolic and end systolic volume. We'll talk about those a little again later, but end diastolic volume is the max volume in the heart. It's the volume of blood in the heart after rest. And end systolic volume is the lowest amount of blood you'll ever have in your heart is after ventricular systole, where all the blood has been forced out. So when do we see a decrease in volume? Right here. As soon as ventricular pressure exceeds aortic pressure, blood starts to leave. It continues to be ejected until right here. Semilunar valve closure. Second heart sound corresponds with ventricular relaxation, the T wave, and that would give our maximal ejection. The difference in volume between this point and this point is how much blood was forced out of the heart in one contraction. Okay? There's a lot here. Please ask questions on Canvas if this doesn't make sense. We will do a quiz on this next week. Okay, so I'll post something on Canvas about this, just a practice quiz, so you guys get an understanding of the way I'll ask questions. So let's move on to cardiac output. We're in kind of the home stretch here. So what is cardiac output? Well, it's this. It's the volume of blood pumped out by a ventricle in 1 minute. Here's what's really interesting. This is going to be the same for both ventricles. Even though we didn't really touch on it actually, unfortunately, I'm sorry, I should have. Let's go all the way back here. I do just want to mention that the right heart has less muscle in its walls because it pumps blood at a much lower pressure. It's just pumping blood to the lungs and back. Whereas the left heart needs to pump blood all the way to your big toe and your finger and the top of your head and back. So it needs to pump at a much higher pressure, much more forcibly. Still though, the cardiac output is actually the same, which is interesting, and we'll talk about that in a little bit. Let's find the slide. Okay, so cardiac output is equal to heart rate times stroke volume. So how fast your heart is beating times, what we're going to learn in a second, how much blood leaves your heart per beat. But real quick, I want to talk about left heart versus right. So which heart pumps blood at a higher pressure? The left heart or the right heart? That would be the left. The left heart pumps blood at a higher pressure. Which heart pumps blood, or which heart has more muscle? That would be the left heart. Which heart pumps more blood per minute? Left or right? You might be tempted to say left on a test, but that would be incorrect. It's both. They both have the same cardiac output in a healthy heart, because these pumps are in series. So if the left heart was pumping more than the right, blood would start to pool out in your tissues in systemic circulation. If the right heart was pumping more than the left, blood would start to pool in the lungs. And these are two different types of heart failure, actually, if you've ever heard of left-sided heart failure or right. So you can kind of guess what some symptoms might be of each. So let's do right-sided heart failure. So if you have right-sided heart failure, that means your left heart has a higher cardiac output than the right, because the right side is damaged for whatever reason. So what might you see? You might see edema. You might see fluid pooling in your tissues because you're pumping blood into the system and it can't get back into the right heart quickly enough because the right heart is backing up because its output is lower. So you would see venous distension. Your veins would get big. You might be able to see their jugular veins in particular really easily, and they'd have chronic edema and high blood pressure. Because when you measure blood pressure, you're measuring systemic blood pressure. Now, what if it was the opposite? What if you had left-sided heart failure? Well, now you have fluid backing up into your lungs, and you might see a productive cough. Fluid cough, because fluid is actually leaking out of your blood into your lungs, which is no bueno. So anyway, that's just an example. In a healthy heart, though, the cardiac output of both ventricles is the same. So that was cardiac output, heart rate times stroke volume. What's stroke volume? Well, we already kind of saw end diastolic volume and end systolic volume. So if you take the difference between the maximum amount of blood, before the ventricles enter diastole, before they enter systole, so end diastolic volume, and then if you take the amount of blood in the heart after a maximal contraction and you take the difference between the two, that equals how much blood left the heart in one beat, which is stroke volume. So let's say end diastolic volume is 120 and end systolic volume is 50. That means your ejection, this will often be represented as a fraction, like an ejection fraction, how much blood actually leaves your heart per beat. But anyway, the stroke volume would be 70 milliliters. Okay, so we need to talk a little, this is going to be crucial to regulating cardiac output, right? Because two things regulate cardiac output, heart rate and stroke volume. Stroke volume is very critical. It's not just heart rate. So what regulates it? There are three things: preload, contractility, and afterload. Preload is the most important. What's preload? Preload is the degree to which cardiac muscle cells are stretched before a contraction. The heart is unique. If you remember skeletal muscle, last quarter we talked about how every skeletal muscle has an optimal resting length. So, and it's usually about halfway. So we use the example of like carrying groceries or lifting a weight. Like if you're doing a bicep curl at the gym and it's hard to start it, but then there's that range in the middle where you feel really strong and then it's kind of hard to finish it. So you like sort of swing the weight to start it and sort of lean back to end it, right? Because there's that optimal strength in mid-range. Skeletal muscle is very strong in mid-range. Cardiac muscle, that's not the case. The more you stretch the heart, the stronger it gets. So the more preload you can put on the heart, the more forcefully it will contract. So increased preload then equals increased stroke volume. And this is referred to as the Frank-Starling law of the heart. The more stretch we can put on cardiac muscle, the more forcefully and fully it will contract. So how do you influence preload then? How do you stretch your heart more? There's really only one way, and that is, sorry, my wife and my daughter just walked in, they distracted me. There's really only one way, and that's venous return. All right, if you get more blood coming back to the heart, that increases preload, and that's perfect because that just so happens, you just so happen to increase venous return when you start to exercise, when you start to contract muscles. Our veins are called capacitance vessels, which means they hold a large capacity of our blood. We may have 70% of our blood in our veins as you sit and listen to this right now. But if you jumped up and started doing jumping jacks or air squat or jump squats or whatever, you're contracting these muscles, especially the ones in your legs. And that's forcing the blood out of your veins back to your heart. So you increase preload, so your heart starts to stretch and beat really hard and more quickly as well. You get your heart rate up. So therefore, cardiac output goes up because you're increasing stroke volume and heart rate. Contractility is the second. So there are other ways to increase the contractile force at a given length. And that has to do with calcium entry. Your sympathetic nervous system is able to do this. There are these things called positive ionotropic agents. Epinephrine would be the natural one from your sympathetic nervous system. There are prescription ones, like digitalis, and then glucagon, which is a hormone that we talked about last week. These all increase contractility, and that comes from sympathetic stimulation. Or glucagon would come from low blood sugar, basically would cause glucagon release, or digitalis would be prescribed. And then lastly, we have afterload. Afterload plays a pretty minimal role in determining stroke volume, but this is the pressure that the ventricles must overcome to open either the aortic semilunar valve or the pulmonary valve. So basically, if this is 140, I'm sorry, it would never be 140. So this would be your diastolic blood pressure. So let's say it's 80. That would be a healthy diastolic blood pressure in systemic circulation. You need to generate 81 millimeters of mercury to open this valve. And it wouldn't open very wide with just a 1 millimeter mercury difference, but you get what I'm trying to display here. If your diastolic blood pressure is 100, now you need to generate 101 millimeters of mercury to open this valve. So hypertension causes your ventricles to have to produce more force to open these valves, and therefore your stroke volume would go down because you're not getting as much blood out with each beat. So hypertension negatively affects stroke volume. And in response, your heart will enlarge to overcome that. And that's kind of that vicious cycle of hypertension and cardiomegaly and heart disease. But anyway, so definition is the pressure that ventricles must overcome to eject blood. So the heart doesn't need the nervous system to contract, but it is regulated by the nervous system, the autonomic nervous system in particular. So this is the most important extrinsic control mechanism. The sympathetic nervous system increases the slope of pacemaker cells, bringing the depolarizations closer together, which increases the speed of your heart rate and increases contractility. The parasympathetic nervous system does the opposite, and most people's hearts display something called vagal tone, meaning that the parasympathetic nervous system has more influence over the heart. It's actually slowing the heart down versus the sympathetic nervous system speeding it up. Now that changes based on situation. If you're exercising, if you're stressed, if you're in a fight or flight response, but typically your heart, your SA node is being slowed down by the parasympathetic nervous system. And if you're in great shape, that becomes even more dramatic. That's why your heart rate lowers as you are in better and better shape, better aerobic shape. And guys, I can't believe it, but that is it for the heart. So I'll get this posted here in the next few minutes. You can go on Canvas, ask any questions you want, and I will shortly, probably Monday or Tuesday, be posting a quiz on endocrine from last week. And you guys can take as much time as you want to do that. Well, let's give it a week. Okay, so you have a week to do it once it posts. And then I'll probably put some heart questions in there too. I know you just saw this, but again, you have a week to do the quiz, so it shouldn't hopefully be too stressful for you guys. Anyway, thanks for watching. Talk to you guys soon. Bye.